Okay, thank you very much, Dr. Lee, Dr. Jia Lee, for joining us today. So, just a brief introduction about Dr. Jia Lee. She's a reader in biological chemistry at Imperial College London. She graduated from Chemistry Department, Nankai University, China in 2005 and obtained her PhD in biochemistry from the University of London back in 2009. Dr. Lee has been awarded many prestigious fellowships, including Imperial College Research Fellowship, Medical Research Council New Investigator Grant, and a European Research Council Starting Grant. Her research team investigates the complex host-gut microbiota-diet interactions and the function of these metabolic crosstalk in modulating metabolic health and colonic cancer. She has a wide range of multidisciplinary research skills, including NMR spectroscopy and LCMS-based metabolic phenotyping, gut microbial profiling, microRNA, RNA analysis, cytokine profiling, and multivariate statistical analysis. We're very privileged to have Jia here. Jia, all yours. Thank you very much.
Thanks very much, Professor Sadiq, for your introduction and your invitation for me to present here. So I don't, just to check, can you see the little video screen on my shared screen?
We can see your shared screen and you as well.
Okay, that's good. That's fine. Yeah. Okay, good. I guess good afternoon, everyone. So for me, it's still in the morning. And today we're going to talk about host gut microbiota and diet interactome. So as you, that's the first technical issue. Oh, that's a flip. Okay, good. So these three components are highly interact with each other. As you know, our diet gets into the our gastrointestinal tract. It actually interact with the microbiota. It provides nutrients to the gut microbiota. In a way, the gut microbiota can also produce a lot of different metabolites, and these metabolites can influence the host immune system or metabolism, the whole host phenotype, essentially. And of course, the diet can, some of the components can be directly absorbed into our body. And to study these complicated interactions is really important in terms of personalized medicine. I hope I can convey this idea to you in this talk. So, as you probably know, the gut microbiota has been shown to be associated with many different human diseases, including some local diseases, as in like colon cancer, and inflammatory bowel disease. This is also in the gut, but also associated with some distant diseases like neurodegenerative diseases like autism, Alzheimer's disease. They're also associated with some systemic diseases such as obesity and diabetes. So it's really important to look at the function of the gut microbiota. And our gut microbiota is not always stable. In the first 3 years of our life, the gut microbiome starts forming into a sort of a consistent composition. And it varies a lot in the first 3 years, but gets stabilized. And then in the elderly, this sort of composition becomes a bit more variable again. But throughout our life, there's lots of different factors modulating our gut microbiota. This includes, method of delivery, the birth, how we were fed as a baby, and our diet throughout our life, which I'm going to mention a few today. And also different diseases, medications we take, like antibiotics, environment we live, what we expose to, what lifestyles we have. And recently, there are also papers showing the host circadian clock also can influence the gut microbiome composition function. So the question is, can we control those factors to maintain a balanced function of the gut microbiota in order to keep our health? So I do believe this is a yes. However, the host microbial and diet interaction is very complex. So as you can see from this figure, the blue bubble here demonstrates the gut lumen, and you can see lots of different metabolites coming from our diet. They can interact with gut microbiota, for example, choline that we take from, for example, meat and egg. They can be metabolized by the gut microbiota into trimethylamine or TMA, and that can be oxidized, taken into the body, oxidized in the liver to trimethylamine and oxide or TMAO, which has been shown to implicated in some of the cardiovascular diseases. So that's just one example, but there are 1,000 different metabolites present in our lumen, and I believe all of them would play a role in our health and disease. How do we actually look at those metabolites? So one of the really well-established methods is called the metabolic phenotyping. This is defined by Professor Jeremy Nicholson a couple of decades ago. And you can see here is a quantitative measurement of multiple samples, essentially, or responses that we measure in the sample from a complex system. So when we actually give a stimuli to a system, or say, for example, a cell, and the cell can respond to it and also change their metabolism to cope with this stimuli. And what we do is to measure those metabolism in order to find out how the cells respond to different stimuli. In terms of methodology, exactly what we use here to look at metabolites. So you can see here, typically we use two platforms. One is called nuclear magnetic resonance spectroscopy, or NMR. This is very stable in terms of sort of stability of the samples. If you run a sample today and run the same sample, say a week or a month later, it gives you exactly same profile. So it's very good when you have a longitudinal studies you wanted to run over years. And then the other platform is called the mass spectrometry. That's more sensitive, and you can couple the different separation methods, for example, liquid chromatography or gas chromatography in front of the mass spectrometry. So you simplify the biofluids or your sample, and then get a mass to charge ratio of the molecules present in your sample. This is very sensitive, as I said, compared to NMR. However, the stability is low. So reproducibility, if you run samples in batches, you are likely to see the batch-related variation. However, there are ways to correct for it nowadays. And this is a sort of example of how the spectra look like. This is NMR spectra, and you can see a lot of different peaks that tells us what metabolites are present in there. So similarly, this is a mass spec spectra data. It's the same thing. Each of these peak represented mass to charge ratio, and the intensity is corresponding to the concentration, but not exactly in terms of mass spec. There are other factors can influence this intensity, so it's not linearly correlated with the concentration of metabolites in your sample. So as you can see, this spectrum is really complex. What we do is to use a machine learning approach to analyze those data first. So typically we can use the principal component analysis, or PCA. So it's unsupervised analysis, meaning that you put a bunch of samples into the program and you ask for the basic intrinsic pattern in your sample, in your data. And it doesn't really need the information in terms of which sample is coming from which group of the experiment. And then after we do the unsupervised analysis, we often perform a supervised analysis. So in this way, we tell, okay, this group of samples are coming from control, this group of samples are coming from, say, patients with IBD, and then could you tell me the differences that contribute to those classifications? And then we sometimes also do correlation analysis or association analysis, so we can integrate different data sets coming from the same sample, same patient cohorts. So typically we do correlation between metabolites and the gut microbial composition, or metabolites with RNA-seq data, for example. So just diving a little bit on the methodology. So this is NMR or different platforms as I introduced earlier. So these platforms are really robust in terms of the sample types that can take. You can see there's all different kind of samples you could put into those instruments. Typically, we use urine, blood plasma or serum, fecal water or feces, and biopsy tissue. So this is very typical for type of examples we use in the clinical setting. But when we do some mechanistic studies, we also use medial cell extracts or either biofluids. For example, recently we look at the saliva, breast condense, and so on. And also actually those platforms are applied to look at the plant metabolites composition. So this is, I would say, it's a completely different field, because the plant have very different sets of metabolites. It's much more complex. So there's also metabolomics used in plant metabolism investigation. But nowadays we're actually trying to merge the two, because in our diet it's quite a lot is actually plant-based. So metabolomics has been used to look at the dietary composition in terms of what chemicals are present. So why do we use NMR or mass spec as the typical tool, and why do we use biofluids? The reason is that you can see this a typical example of NMR spectra of urine. And these peaks telling us information about endogenous metabolites that's present in us, and also tell us what we have actually eaten. So the metabolites present in diet, as well as metabolites produced by our gut microbiota. And on top of that, it also tells us the metabolites derived from this interaction, for example, host microbial co-metabolites. So it means that those set of metabolites are produced by the collaboration between the gut microbiota and our host system. So the example I gave you early on, TMAO, is a typical host microbial co-metabolite. And also our metabolome is very unique. It's like our fingerprint. This work done by my previous PhD student, Dr. Juterop. So he's now back to Thailand, and also an academic faculty position, working on metabolomics, setting up a Phenome Center in Thailand. So what he did was to collect samples from 5 healthy participants over a 6-month period. So we took 1 sample per month. And when you do a PCA analysis unsupervised, and what we see is actually so here each color is 1 donor. You can see each of the persons actually forming a little cluster here. The middle ones is clustered together. And if you zoom in that and redo a PCA, you can see, again, this is 3 people more or less separated. We see these red and blue dots that clustered really close to each other, because they're actually both a male British Indian background. So maybe their dietary habits is quite similar. But essentially what I wanted to make a point is that you can see their metabolome is quite unique. And if you look at their fecal bacterial composition from here, and you can again see a very distinctive clusters down here, meaning that their microbiome, particularly the bacterial composition, is very unique for each other. And it's quite stable, I would say, across the 6 months. And those participants didn't have any antibiotics in this period. So antibiotics, like I mentioned earlier on, medication can change our gut microbiota. And antibiotics is a very extreme example obviously you can wipe out a fraction of the gut bacteria if you take it orally. So here, this work we done many years ago in mice. We give mice some a dose of vancomycin, and then we followed up their metabolic shifts over 3 weeks after a single dose. So you can see, again, this is a 3-dimensional PCA plot. Here, each dot represents one sample and color-coded based on the time points. And again, you can see after 1 to 3 days after the vancomycin treatment, those profiles shift away from the control and the remaining time points. And same thing you see from the urine spectra. Here we have 3 time points, 1 to 3 days post-treatment. And quite a lot of metabolites have shifted, which causes this clustering you see in the PCA scores plot here. So that was just an example and some background information about this field. And now I'm moving on to a few examples I'd like to show you. So first, I would like to start with nutrition. So I want to show you 2 studies we've done previously. One is a kwashiorkor as a sort of acute malnutrition condition, and we have used the peanut-based therapeutic food, or so IUTF diet to do the intervention and followed up their metabolic shifts after the intervention. And the second study is about colorectal cancer with a high fiber diet. So with this kwashiorkor study, this is in collaboration with Jeffrey Gordon, who's a well-established scientist in microbiome, one of the pioneers. So he has done a sample collection from, I believe this is in Malawi. So he had collected samples from the children who are twins but discordant for healthy and kwashiorkor. So here it has over 300 twin pairs, and have selected the same gender nourished twin pairs, so 9 pairs, and they also selected the 13 twin pairs which are discordant for those conditions. And they followed them up for 2 to 3 years, and also IUTF diet is given to the kwashiorkor, give those twin pairs. And then they look at the fecal samples in terms of their microbiome composition and function. And you can see here, this is a typical sort of correlation between the age and the variation in the microbiome data. So like I said early on, the first 3 years of our life, the microbiome has a shift. Well, it's shifting a lot because we have, weaning period, and we have different diets, and we change from soft food to more solid food. So that's the biggest influence in the gut microbiome composition. And you can see this very clear here. Regardless whether these twins are kwashiorkor or healthy, what you see is is a very nice correlation between the age of those children and the microbiome composition. But if you split the composition, if you split the healthy versus kwashiorkor twin pairs, and you can see their sort of microbiome shift along the time. So the top panel here is the 2 twins who are all healthy, and you can see the changes in the microbiome is very similar along the time. And the bottom one is the discordant twins, and the left is healthy. You can see it's similar to the healthy twin pairs. But the discordant ones, you can see even after IUTF diet, their trajectory of the microbiome shift is different. And then we thought, okay, maybe we take those fecal samples and transplant into the germ-free mice, and to follow up to see how exactly those microbiota responds to different diets, and how exactly their metabolome respond to those diets. So that's the study design. We have a healthy and kwashiorkor twin fecal donor put into the germ-free, and we give them a lowering diet for 19 days and followed by 2 weeks of RUTF diet, and switch back to the Malawian diet for another washout for another 4 weeks. And then we collect fecal and urine samples. So here again, we look at the microbiome first, and you can see here the blue ones are coastal recipients of the mice. You can see their microbiome shift, very distinctive to the healthy ones when we give them RUTF diet. They sort of dropped here. And if we look at the microbiota composition shift here using the heat map, so you can see here during the RUTF diet, the middle panel here, the bifidobacteria has increased, and the ruminococcus is also increased together with the lactobacilli. But after you shifted to Malawi diet, you can see all of those changes sort of disappeared. So those diet-related microbiome shift is very short-lived. We next look at the metabolome. So again, you can see the urine. You see differences related to the diet. So RUTF diet is separated from the Malawian diet. Then we remove the RUTF diet. Just look at the 2 Malawian diet treatments before and after the RUTF diet. And what you see here is the differences between the solid dots, which is healthy microbiota recipients, versus the coastal recipients. So that means after RUTF diet, the diet doesn't really induce persistent changes in the urine profile. So once you remove the treatment diet, the host metabolic status shifts back to where it was. That means RUTF diet, once you stop, the therapeutic effect is probably quite minimum. So this is just a main message of this study. So like I said, we proved that gut microbiota could be a causal factor in coastal, and RUTF diets can change the microbial and the metabolic profiles, but those changes are really short-lived. The next study is about diet and colon cancer. So colon cancer is a third commonly diagnosed cancer in both male and female. And across the world, the African Americans have much higher colon cancer risk compared with Native Africans, given that they have a very similar or same genetic makeup. Then we hypothesized that this difference is due to a Western diet. So American diet increased the risk of colon cancer. So to test our hypothesis, we recruited 20 healthy African American participants as well as 20 Native Africans. So we let them to have their own diet. For 2 weeks, we collect the samples, and then we swap their diet for another 2 weeks and collect the samples. And just to give you an idea what's the difference between the two diets. So in Western diet, we have lots of carbohydrate, but this is mainly due to the sugars. Also, we have a high fat composition. In contrast, in African diets, we also have a lot of carbohydrate, but this is mainly fiber, and we have much lower proportion of the fat, and calories also lower. So we took our biopsies from those participants at the end of the study. So what you actually see here is three different biomarkers that can be used to assess the colon cancer risk. One is the Ki-67 that represents the cell proliferation, and CD3 and CD68 are 2 inflammatory biomarkers. So if you look at the blue box here is the Africans. Is the African Americans. So you can see here when they had the African diet, those markers reduced, meaning their colon cancer risk is reduced. Whereas if you look at the Africans, when they had Western diet, all of these risk markers is increased. This is just the 2 weeks, meaning that 2 weeks of dietary change, it can modulate our colon cancer risk biomarkers very quickly. And we look at the gut microbiome. Surprisingly, that the bacterial composition didn't shift dramatically. It's only some minor compositional shifts we observe. However, it's the connection between the gut microbes. So we can see a occurrence of some butyrate produces Utrobacterium rectale. They increased, and this increases together with some other bacterial that's associated with or have a function of metabolize the complex carbohydrate. So those gut microbiota have become more active. That's probably because more of the fiber has been fed into the colon. Then we look at the metabolome, so very consistent to what we see in the microbiome. Because this is Africans, greens their home environment, and blues after Western diet, they shift dramatically there. And the shift is due to some of the branching amino acids. We know those associated with type 2 diabetes. We also see the shift in TMAO, which is related to cardiovascular diseases. And then you can see Americans, when they had the African diet, they also have a shift in their metabolome. But majority of those shifts actually are the metabolites coming from vegetables like broccoli. So this group of metabolites. So that's again showing that metabolic profile can reflect what we eat. And the next, we use some complex network analysis. You can look up this bioinformatic tool from the paper from Joran Postma. So I just wanted to highlight two things here. One is in the Americans after African diet, you can see their aromatic compound metabolism is increased, whereas in the Africans after American diet, their glycolysis and sugar metabolism is increased. So message from this study, I think is quite brief. So having a healthy diet is definitely good for your gut. But essentially what we see is that African diet that high fiber, low fat can reduce the colorectal cancer risk, even just within 2 weeks. So I don't know how I'm doing with the time. So just want to show you next part of my work is to do with obesity treatment, mainly the bariatric surgery. Bariatric surgery is also called weight loss surgery. You can see it can, there's 3 different types of surgery here. Gastric banding, just put a adjustable band on top of the stomach. And the second is sleeve gastrectomy, which is becoming popular now. That's removing 3/4 of the stomach. We call it restrictive procedure. And the Roux-en-Y gastric bypass is gold standard, but now less people are having this operation. It's very complex. So you first restrict the stomach side. Secondly, you bypass the duodenum and connect the end of duodenum to the lower part of the jejunum. So it creates this Y shape. So food coming down from here, you can see bypass the most of stomach and also bypass the duodenum and go down to the small intestine. Those surgeries not only effective in terms of reducing excessive fat, but also can improve type 2 diabetes within weeks. So we have done a study here in humans. So we have included a 4 group of patients. One is having RYGB surgery. Second is having very low calorie diet, which is matched to the calorie the RYGB patients have. We also had a gut hormone treatment. So we know after RYGB surgery, gut hormones change. Those are the satiety hormones that change dramatically. And one of the hypotheses for the RYGB surgery is that the gut hormone is contributing to post-surgical outcomes. So here we have this group of patients to control for the gut hormone-induced changes. And we have also a saline group as a control. So after 4 weeks of those intervention, we collected urine fecal samples and plasma samples from those patients. We analyzed using both NMR and mass spec techniques, and those followed by the multivariate statistical analysis. This is a PCA score plot, and see each dot here represent one sample or one patient color-coded based on their treatment. So main message here is that the RYGB patients in purple is very different from the other groups, and a very low calorie diet also induced quite a lot of changes. This is mainly the ketone bodies. When we starve our body, the ketone body produced more. In RYGB surgeries, mostly microbial co-metabolites increased. And we can't really see any differences between the gut hormone treatment groups and the saline control group. Probably indicates gut hormone treatment doesn't really induce quite a lot of metabolic shifts in the urine and feces. So our second studies, we focused on the RYGB patients. So here we followed them up for a bit longer. We have 68 patients to start with, and we took sample at 2 to 6 months post-surgery, as well as 1 to 2 years. And similar methodology has been applied to those samples. So first thing first, we can see a significant body weight drop in those patients along the whole year. And when we look at the urinary metabolic profiles, so here is you can see black is the pre-surgery, and orange and green are the post-surgery at 6 months and 1 year. And you can see a roughly clear separation between the pre and the post-surgery time points. And the picture, the figure on the right, it's a bit more complex, but essentially these are the sort of NMR spectra. Here, the red peaks, meaning they're the important metabolites contributing to this classification. Direction means how they change between the two groups. So we can see those red peaks here, they indicate some of the host microbial co-metabolites, for example, phenylacetylglutamine, doxysulfate, paracrysal sulfate. They all increased after RYGB surgery at both time points. And similarly, we look at the feces, you can again see differences between all of the time points. And if you look at the loadings, which is metabolites that contributed to those classification, it mainly increased the phenylacetate here and also tyramine. If I have time, I'll mention this metabolites later on. So again, we look at the bacterial composition. Again, we see a similar classification as in urine and feces of their metabolome. And then if specifically we look at which genus is changed, you can see Klebsiella and Escherichia, they all increased after surgery. And those bacteria are not usually a sort of a healthy bacteria, they're potential opportunistic pathogens. Then we wondered whether bariatric surgery is actually a good option for patients with obesity or not. Then we did a statistical correlation between the bacteria and the metabolites. As I mentioned early on, this is a very typical correlation approach we use to sort of give us an idea what metabolites are potentially linked to which bacteria. So you can see here, tyramine and PEG, phenylacetylglutamine, is correlating with a bunch of bacteria down here. But we have to be careful. This is statistical correlation. It doesn't mean those bacteria are producing those metabolites. So we next is to take a fecal sample from healthy donor and a RYGB donor, and we cultured them for 24 hours, and then we give them different substrate to see if those bacteria actually have different metabolism or not. So here just wanted to show you example of Tameer-e-Millat. So as mentioned, they increased in bariatric surgery. The precursor of tyramine is tyrosine, is an aromatic amino acid. So when we give these bugs tyrosine, the healthy donors bugs didn't really convert tyrosine into anything, but the RYGB bacteria has converted tyrosine into tyramine, which probably caused the increased tyramine in the RYGB fecal samples. And again, so we did some other bacterial culture as well, and similar thing, we can see different bacterial that are present in the RYGB patients, metabolizing some of the precursors of host microbial co-metabolites PEG I mentioned earlier. The precursor is phenylacetate, which we saw to increase in the fecal water after RYGB surgery. And again, if you culture the bugs from RYGB surgery, you can see these bugs are able to produce more of the phenylacetate. So I think I may skip this bit because I think I'm running out a bit time. So essentially wanted to say the RYGB metabolism also carries on when those people are becoming pregnant. You can see this is pregnant mothers. These host microbial co-metabolites are consistently higher in those pregnant women compared with non-operated pregnant women. And surprisingly, in their baby, some of those co-metabolites are also present in higher levels compared with the baby born to mother without surgery. So I just wanted to highlight the last part of my work. So what I've shown you early on is mostly sort of observational. So we look at how the global metabolic shifts are changing, and we look at how the bacterial composition is changing. We do statistical correlation between them. But essentially, I really wanted to ask one question. When those metabolites are increased or decreased, what do they do to us in terms of a chronic health? So in the last probably 3-4 years, I'll be focusing on a few metabolites function in terms of their colorectal cancer in developing of colorectal cancer. So tyramine, I have mentioned a few times here. It is a metabolite coming from bacteria by metabolizing tyrosine. But also we can find tyramine from different food, which is fermented. So also bacteria present in those food. You can see fermented fish sauce, cheese, ham, cured meat. So tyramine also recently, I think early this year, the paper published reporting that tyramine is associated with the inflammatory bowel disease severity. So we thought maybe tyramine is related to inflammation, which also increases colorectal cancer risk. So this work has been done by Dr. Glymanaki. So she had first looked at the tyramine effects in the human chronic epithelial cell lines. This is HCT116 cells. So when we give them tyramine treatment at different doses, you can see from the 1.2 millimolar, the cells started to develop the genotoxicity, and this is measured by micronuclei. So you can see a little micronuclei formed here in the cells treated with tyramine. And we also used another marker to look at the DNA damage and repair. So this is gamma H2AX, again, very consistent. You can see here at concentration above 1.2 millimole, the cells are starting to have increase of this marker. As I mentioned before, cell proliferation is another colon cancer risk biomarker. And when we look at Ki67 across different concentration, also at 1.2 above, the cells started to proliferate more. So this evidence sort of promoted us to start an animal work. So we took APC mean mice, which is a genetic colon cancer model, and also wild type animals. We give them tyramine for about 7 weeks. We calculate a tyramine sort of concentration based on a human intake of, say, 200 grams of ham if you eat, and how much tyramine equivalent in there. So after 7 weeks, we looked at their tumor development. So you can see here, this is APC mean mice treated with tyramine. They present more tumors compared with the APC water treated. And surprisingly, we actually see some tumors in the wild type animals as well. I don't think, well, I don't got data show here, but we did see a small fraction of animals from a wild type also developed tumor after tyramine. And then in the cell proliferation of the colon of the intestine, we can also see that the APC mean mice fed with tyramine had increased the cell proliferation compared with the control mice. And you can see also there's a tendency increase in the wild type animals, but it's not statistically significant. And genotoxicity. So again, we can see this genotoxicity actually increases significantly in the wild type animal treated with tyramine. We can't see this as a huge significant differences in APC mean mice, but I think, you know, in those genetic model, the gene mutation is the main driver for tumorigenesis. That's probably why we don't see the significant differences here. So we also did a little bit organoid work here. So we cultured the small intestinal organoids from wild type mice, and we have this control organoids, and these are the organoids treated with etoposide dissolved positive control, and these are treated with tyramine. And you can see those purple dots here. These are representing the DNA damage induced by the tyramine, but you don't really see this in the negative control organoids. So I think collectively those evidence demonstrated that tyramine can increase colorectal cancer risk factors, as well as promoting the development of tumor growth. So just wanted to finish my slides by presenting these big ideas of precision medicine. So you can see here, this is our ultimate goal. I hope I've shown you that the potential of using metabolomics to actually achieve this goal, because metabolomics can detect the changes in our microbiome, nutrition, lifestyle, and hopefully those data can be integrated with other omics data like genomics, transcriptomics, and proteomics to link together to look at how our body responds to different stimuli, and hopefully that can achieve precision medicine one day. So finally, I'd like to thank people who have contributed to those work I presented today, as well as the funding bodies who provided the important funds to support those. And thank you very much for your attention. I hope I make it within the time. Thank you.
I was mute, sorry. Thank you very much, Gia. It was wonderful talk. It's just that where I'm sitting, the internet connection was unstable, so I was like, you know, I was having a bit of a trouble. Anyway, Gia, there have been so many questions, so I hope we have a few minutes. If I could start with my personal question, just for the interest of the audience. You know, when we eat anything, like our gut microbiota changes, it reflects very quickly. So what is the time, you know, duration? Is it within 24 hours, 48 hours that our microbiota changes? And what if, you know, if there are any, if somebody has, you know, Lynch syndrome or something in colon cancer, for instance, or somebody has pancreatitis or, you know, a chronic condition?
Yeah, that's a very good question. So I think when we talk about microbiome, we actually refer to both the compositional shift and the functional shift. So the compositional shift, as in what bacteria are there, how many of them are there. I think these changes slightly slower compared with the metabolism changes. So I think, you know, after 24 hours, we see the metabolomics changes in our urine feces. So I would say, yes, it's very rapid in terms of their functional changes. Compositional changes may take a bit longer. Whether the patients have a similar rate of changes, I don't know. I believe yes, but I don't have the data or come across any papers looking at this.
Right. You know, you did mention like African diet, like high carbohydrate, low fat diet is good. Actually, Pakistan also has similar sort of diet, but here early onset of cancer, colon cancer is increasing at an alarming rate, you know, like girls, boys, teenagers, very young, you know, they're coming up. So, you know, we're into that research, like we want to look into the factors that are contributing to that early onset cancer, having, you know, even after having high carbohydrate diet. What else do you suspect, you know, could be the reason? Microbiota to play a role, or?
I think, you know, this is actually a global observation. I think there's a paper showing globally colon cancer is reducing the incidence. However, it's increasing the incidence in the young people. People do question, you know, what exactly actually contributing to this. I think diet is the major. I believe diet is a major factor here. You know, the babies nowadays exposed to the diet is very different from, you know, the diet exposed to, say, 20 years ago. And that early life exposure probably play a very important role in their later on life health. And also this westernization, I would say, you know, like McDonald's, all of this sort of high energy food is, you know, you find it globally. You know, even at home, we eat our traditional diet, high fiber, but probably the kids actually exposed more of the Western diets compared with the kids 20 years ago. So that might be another reason. Whether gut microbiota can play also a role there, I don't know. I think transgenerational effect is really important to look at. That's why we recently just trying to submit a grant actually today on that to look at how the gut microbiota from you know, the bariatric surgery patients influence their offspring. You know, we do believe there is a transgenerational impact there, but we haven't really studied it yet.
Right. Let's wait, you know, until you get the grant and you carry on working on this. So I did ask, you know, this a bit of this question, but, you know, I can repeat that. Somebody's asked, what changes occur in microbes of patients with recurrent or chronic pancreatitis, any chronic inflammatory conditions? What can we do to maintain gut health in chronic conditions?
Oh, wow, that's a very big question. I think we haven't found a bacterial marker that's linked to chronic inflammation. I mean, this is excluding acute bacterial infection or whatever, but there isn't a consistent sort of bacterial species, and we know that induce chronic inflammation. I think chronic inflammation is such a complex event. So it's not, I don't believe it's caused by a single bacteria or a single metabolite. It's probably caused by a prolonged exposure to certain pattern of metabolites. And I think our body has resilience. So when we are healthy, we are able to cope with sort of a harmful stimuli.
Yeah.
But depends on how long we expose to this harmful stimuli, then we lose our resilience gradually. So I think come back to the question,
Yeah.
you know, what metabolize, how we maintain this. This really depends on sort of individual level. Come back to personalized medicine, personalized nutrition. This is probably quite personalized. And I think people have taken, for example, probiotics to maintain, trying to repopulate the gut microbiota, balance it, maintain a good health. But that does work for some people, but don't work for others. And we don't know why. And in our study, we did in, I think, Cardiff, or a few cities in the UK, we actually didn't see any changes in those population after 6 months of probiotics treatment. So we don't know. I think it does need more evidence, and does need more detailed evidence on individual people.
So, Gia, yeah, you did mention antibiotic exposure in your talk a bit. Somebody's asked, how do environmental factors such as antibiotic exposure, stress, and lifestyle impact the host gut microbe diet interactome? The thing is, Pakistan has a very high antimicrobial resistance as well. So, you know, what do you say?
Sorry, are you asking how do you keep how to
how does
factor after the treatment?
Yeah.
Okay.
How does it affect? Yeah.
Great question.
Yeah.
Yeah. So there are papers showing that there isn't a best way to maintain or to repopulate the gut microbiota after antibiotics treatment. So perhaps promoting a diverse gut bacterial, one way is to give a diverse range of food. So if you feed the bacteria different food that they need, there might be a way to pump up their population. There are studies that done, I think, published in Cells a few years ago. They have done antibiotics study in human. They gave them probiotics, but proven that probiotics was not able to repopulate the gut microbiota bacterial composition very well. Similarly, we have done this in animals, in rats as well. We also transplanted the rats' feces, fecal bacteria, after antibiotics treatment. It still doesn't work very well. The best way is to let the rats to recover their own bacterial for 3 to 5 weeks. So yeah, I mean, I don't know, but I think giving them a comprehensive and balanced diet is probably the one of the options.
Yeah, diversity in food, that really counts. Just to regain a healthy gut microbiome. Just, you know, like there are quite a few questions and all around, you know, like similar sort of theme. So I don't know. Just out of my curiosity, you know, Pakistan has like almost 30% of our population has diabetes. So that's another thing, you know, what do you think? Like, on one hand, you should have high fiber diet for healthy gut microbiome. And then if the like 30% of your population has diabetes, you know, doctors would rather advise them to have like more protein than carbohydrate. So where is the balance? You know, in your opinion, what could be done diet-wise?
That's a challenging question. Yeah. I mean, I don't have an answer to it.
It's okay. It's just a food for thought, you know?
Yeah. I don't know. I think probably resistant fiber might be a bit better option. But I don't really have enough knowledge on that to be able to give you an answer.
What is that resistant fiber? Sorry, I don't know about that.
Yeah, so those resistant fiber, meaning that we can't utilize them at all. It's only the gut bacteria can digest them. So in our upper
Mm.
GI, we don't really have that many bacteria present. There are some, but mostly they're in our colon. So when we have those resistant fiber, they would travel down to the colon because we can't use them. And then those bacteria will utilize them in the colon. Whereas if it's a sort of a starchy thing, you know, we can actually digest them, so it will derive lots of sugar and stuff that might be, you know, have an impact on the diabetic, the patients with diabetes. Yeah.
And that's interesting. Actually, we need to look into that, because, you know, like if we want to work on precision, like personalized microbiota, then we need to, you know, like doctors should start advising about that resilient sort of fiber for the diabetic population. But we need to look into that. Yeah. So I think like our time is nearly up. Thank you very much. Yeah, it was wonderful to have you. And I hope to have you again sometime in the future once you have a few more, you know, like hot areas to share with us. Thank you very much. Yeah. Thank you once again. Thanks.
Thank you very
Bye.
much. Bye.
Thanks. Bye. Bye.